Welcome back to the deep dive. Today we are really immersing ourselves in a piece of conservation research.
Yeah, one that perfectly shows how technology is, well, becoming absolutely essential for wildlife managers. It's a really clever study.
We're looking at a paper that takes us right into the world of amphibian monitoring.
It is. For those of you listening, if you want the key insights without wading through, the dense academic text, we're diving deep into effective amphibian conservation monitoring and habitat assessment using eDNA.
Hmm.
The lead author is Matthew Seymour, published in 2025.
Right. And our mission today is pretty straightforward. We want to fully unpack this research for you.
What they looked at, how they did it.
Exactly. The methods, the sometimes surprising ecological findings, and what it all really means for conservation strategy. We want you to feel like you've got the core of the source material.
And the star of the show, so to speak, is the European green toad, Frösäter viridis.
Mm-hmm.
This species is facing, well, enormous challenges, especially in Sweden, where it's actually the most threatened amphibian.
Wow.
Yeah, it's surviving only in these really fragmented populations way up north.
And I know people have put a lot of effort, a lot of resources into trying to save it. You often hear about moving animals, these translocations. Can you just quickly define that?
Sure. So translocation here means the controlled movement and release of animals that were bred in captivity.
Okay.
In this case, it was hundreds of thousands of green toad tadpoles and toadlets. They released them into new or restored habitats in the wild.
Right, hoping to start new populations.
That's the hope. But the problem, specifically for this toad, is that the adult return rate is incredibly low.
Ah.
And the traditional ways of monitoring, listening for calls, setting traps that are just too slow, require a ton of effort. And honestly, they often completely miss these really elusive, low-density populations. Amphibians can be tough to find.
So we needed a serious sensitivity upgrade for monitoring.
Definitely.
And that's really where this technological breakthrough, environmental DNA or eDNA, comes into play.
Absolutely. So eDNA is basically the genetic material that an organism leaves behind in its environment. Things include skin cells, mucus, waste.
Stuff just floating around.
Exactly. If the animal lives in water, that DNA gets suspended in the water itself. We can just take a water sample, isolate that DNA, and boom, you can confirm if the species is there or was recently without ever needing to see or catch the animal.
But the researchers here went a step further than just a basic DNA test, right? They used something called eDNA metabarcoding.
Yes, and that's a crucial distinction. Metabarcoding uses high-throughput sequencing. Basically, it analyzes all the different vertebrate DNA fragments in that single water sample at the same time.
Okay, so not just looking for the toad.
Not just the toad. It gives you this incredibly comprehensive sort of ecosystem-level snapshot of the whole vertebrate community, fish, birds, mammals, other amphibians, all from one sample. It's way beyond just tracking one target species.
That's powerful. So armed with this tool, the Seymour team had three main goals across these Swedish regions, Öland and Kalmar. What were they aiming for?
Okay, first, they needed that definitive proof of concept specifically for the green toad. Could eDNA rapidly detect Frösäter viridis presence right before, and then crucially, immediately after a big translocation effort in 3 specific ponds?
So testing the speed and sensitivity right off the bat.
Precisely.
Yeah.
Second, they wanted to leverage that same metabarcoding power, use it to assess the wider biodiversity in ponds across the region, and importantly, identify potential biotic interactions, figuring out who's living with whom. They surveyed 37 sites for this part.
Right, getting the ecological contact.
Exactly. And third, this leads straight to the practical application. They wanted to use all that data, the species lists, the biodiversity info, those ecological interactions they uncovered, to build a framework.
A framework for what?
To decide which of the surveyed sites were actually the most suitable for future translocation efforts, making those efforts more likely to succeed.
That's a fantastic progression. From checking the tool works to gathering broad ecological data, right into practical conservation planning. Okay, let's dig into the how. How did they set up that experiment to test the speed of eDNA detection?
Yeah, the setup was quite neat. The timing was everything. So at those 3 translocation ponds, this was in May 2021, they collected their baseline samples first.
Before any new toads went in.
Right. Triplicate 1-liter water samples taken 3 hours before they released any of the European green toad tadpoles.
Making sure the ponds were clean of that specific DNA signature first.
Correct. Then they did the release, put the tadpoles in, and collected the second set of samples just 24 hours after the release.
Only 24 hours.
Only 24 hours. They were basically creating this, instantaneous artificial spike in toad biomass to see if the tadpoles shed enough DNA in just one day to be picked up reliably.
Okay. And separate from that specific test, they did the broader survey across, what, 31 other freshwater bodies?
That's right, 37 samples in total across 31 water bodies. The main survey was also in May, during the peak amphibian breeding season, and they were really rigorous about the methods to make sure the data was solid.
How so?
Well, they used the standard 12S meta-barcoding protocol. For anyone unfamiliar, 12S targets a specific region of mitochondrial DNA that's common to basically all vertebrates.
So it's a good scan everything tool.
Exactly. Fish, birds, mammals, amphibians, it picks them all up. And they were clearly super careful about avoiding false positives.
Contamination is always a worry with DNA work.
Huge worry. So they used extensive negative controls at every single stage. In the field when collecting water, during the DNA extraction back in the lab, and during the PCR amplification step.
Checking for any stray DNA.
Constantly. Plus, they also specifically tested every eDNA sample for a major amphibian disease.
Ah, the chytrid fungus.
No,
Indeed.
Yes, Batrachochytrium dendrobatiatus, or BD. They used a highly sensitive technique called quantitative PCR, or qPCR, for that, especially focusing on sites where they suspected infections might occur.
Okay, meticulous. Now, let's get to the results, the payoff. That 24-hour test, what did the eDNA reveal about the green toads right after the translocation?
The results were, honestly, crystal clear, definitive. Before they released the tadpoles, Buphotes viridis DNA was completely undetected in all 3 ponds. Zero signal.
As expected.
As expected. But just 24 hours later, the detection probability for the green toad shot up by 100%.
Wow, 100% increase in just a day.
Yeah, a statistically significant jump, p less than 0.01. It was an unambiguous signal, proving that eDNA can absolutely pick up a sudden arrival of amphibians with incredible speed.
That is staggering efficiency. I mean, think about it. That potentially reduces what could be years of difficult monitoring, waiting to see if any toadlets survive to adulthood down to a single non-invasive test done over 24 hours.
It really changes the game for rapid assessment.
Yeah.
And this is where that metabarcoding power really comes in, even just at those 3 initial translocation sites.
Right. It wasn't only about the toad.
Not at all. From those same water samples collected for the 24-hour test, they also successfully detected 3 other amphibian species.
Which ones?
The great crested newt, the smooth newt, and the common toad. Plus, they picked up multiple species of fish, birds, and mammals just at those 3 ponds.
Okay, so zooming out now to the full survey, all 37 pond samples across the region. The amount of data they pulled from just water samples must have been huge.
It's astounding, really. From that one round of water sampling, eDNA delivered this incredibly rich picture of the entire vertebrate community.
Give us the numbers.
Okay, they detected 8 amphibian species, or genera, including 7 named species like the common toad, moor frog, great crested newt, 15 different fish species, 17 mammal species, and get this, 38 different bird species.
Hold on, 38 bird species.
Yep.
From a liter of water from each pond.
38 bird species detected via their DNA left in or near the water.
That's not just monitoring a toad anymore. That's like running an instant biodiversity checklist for birds and mammals across the whole area at the same time.
It really highlights the sheer scalability and breadth of the method. And, the species richness varied quite a bit from pond to pond. Some only had 1 detectable species, while one location had 24 different species.
Right.
The average is about 8 species per pond.
Amazing breadth. But they didn't just stop at counting species, did they? This massive data set let them look deeper into the ecological dynamics. Who's interacting with whom?
Exactly. And this, I think, is where the study provides really novel, actionable ecological insights. It's the core novelty.
How did they do that?
They use something called co-occurrence analysis. Basically, looking statistically at all the pairs of species, they detected 258 pairs in total to see if certain species tend to show up together more or less often than you'd expect by chance.
Looking for patterns.
Looking for significant associations. And they found some clear negative associations involving amphibians. Specifically, the common toad, the smooth newt, and the moor frog were significantly less likely to be found in ponds where 2 other common species were present.
And those were?
Cattle, Bos taurus, and the gray heron, Ardea cinerea.
Okay, cattle makes intuitive sense. Grazing might disturb the habitat, trample vegetation, or banks.
Precisely. The thinking is that cattle presence might be detrimental, either through that physical disturbance or by altering the vegetation structure in ways that amphibians don't like.
But the gray heron, herons eat amphibians, right? Isn't that just nature? Why is that a negative association that helps you plan where to put new toads?
That's a really important distinction. Yes, predation is natural, but a strong statistical negative association like this suggests something more. It indicates that in ponds, where heron DNA is frequently detected, meaning herons are likely spending significant time there in the world, amphibian populations are less likely to persist or thrive.
Oh, okay.
It suggests that predation pressure might be particularly high in those specific locations, maybe too high for a small, vulnerable, newly translocated population to get established, especially during the summer breeding season when herons are working hard to feed their chicks.
So it acts like a warning sign for managers. Maybe avoid the ponds the herons heavily favor.
Exactly. It turns the eDNA data point for herons into a practical management consideration. Don't put all your expensive captive-bred tadpoles straight onto the heron's main dinner table.
Got it. Okay, that makes sense. Now, on the flip side, did they find any positive associations? Species that seem to benefit amphibians?
They did. They found significant positive associations between amphibians, notably the great crested newt, and a couple of common herbivorous birds.
Which ones?
The mallard duck, Anis platyrhynchos, and the common crane, Grus grus.
Ducks and cranes, helping newts. That seems less obvious. What's the proposed link there?
Well, the hypothesis is that these plant-eating birds actually help maintain a habitat structure that benefits amphibians.
How? By eating plants.
Essentially, yes. By foraging on aquatic vegetation, they might prevent the ponds from becoming completely choked with plants. They help maintain areas of open water, keep the shallow edges clear, basically preserving the mosaic of conditions, including breeding sites and shelter, that amphibians need.
Interesting. So bird activity could actually be a positive sign for amphibian suitability.
It seems so, based on these associations. Okay, before we jump to the final implications, let's quickly cover the disease finding. What did the eDNA reveal about the chytrid fungus, bread?
Right, the deadly fungus.
Yeah, this was a pretty significant finding and maybe a bit concerning. This study provided the first official detection of bread in Kalmar County, Sweden.
That's big news for the region. Where exactly did they find it?
It was detected in 5 specific ponds. And interestingly, it was only found during the spring sampling in May. When they resampled some sites in early autumn, August, they didn't detect it.
Huh. A seasonal pattern, maybe?
Possibly. And perhaps even more telling. Every single one of the 5 ponds that tested positive for bread was a man-made pond, not natural ones.
Okay, that seems important. Did the presence of the fungus seem to be linked to fewer amphibians in those ponds right now? Was it causing obvious declines?
Surprisingly, no. When they ran the stats, they found no significant immediate relationship between whether bread was present or absent and the overall occurrence or diversity of amphibians in those ponds.
So finding food didn't automatically mean fewer amphibians.
Not in this snapshot, no. The p-value was 0.85, so nowhere near significant. It suggests the relationship is probably pretty complex.
What might be going on?
It could depend on the specific strain of food present. Some are more virulent than others. Or it could be local environmental factors like temperature or water chemistry that influence whether the fungus actually causes disease in the Swedish context. It definitely signals that we need a much deeper investigation into food dynamics there.
Okay, complex picture on the disease front. So this all leads us nicely into the final part, the practical use of all this data. How did the researchers take these findings, the negative vibes from cattle and herons, the positive ones from cranes and ducks, and turn it into a tool for conservation?
Right. They used a really powerful decision-making tool called multi-criteria decision analysis, or MCDA.
MCDA, what's that involve?
It's basically a mathematical modeling approach. It lets you assign different weights or importance levels to various factors. In this case, they weighted those key positive and negative influences they'd identified using eDNA.
The ones linked to amphibian presence.
Exactly. The negative factors like cattle presence, heron presence, and also the presence of a predatory fish, the nine-spine stickleback, which they also detected, and the positive factors, like the crane and duck detections.
So they built an evidence-based ranking system for the ponds.
Precisely. The MCDA model crunched the numbers and ranked all the surveyed ponds based on this combination of factors. And it clearly identified 6 ponds that consistently ranked as highly favorable for future amphibian translocations.
What characterized those good ponds?
Typically, high detection rates for cranes and ducks, and limited evidence of the negative influences, not much cattle activity, fewer heron detections, absence of the stickleback.
And
Conversely, the model flagged 7 ponds as consistently unfavorable. These usually scored poorly because of high indicators of cattle disturbance or significant presence of sticklebacks, which are known to eat tadpoles.
Wow. So this really transforms conservation planning. It moves it away from just, this pond looks okay, let's try it, towards a really targeted, data-driven strategy.
It absolutely does. I think the
Yeah.
study beautifully underscores the transformative potential of eDNA metabarcoding.
Yo,
It's not just one tool. It's a single scalable method that gives you both species-specific tracking and that vital holistic ecosystem context.
Yeah, context seems key.
It bridges that critical gap between just monitoring a vulnerable species like the green toad and actually gaining the ecological insights like those biotic interactions that you desperately need to make conservation efforts successful in the long run. It helps us move from just tracking the species to understanding the sustainability of putting it somewhere.
Did the researchers point out any immediate limitations or next steps needed?
They did. They were upfront that for some of the surveyed ponds, they didn't actually have eDNA data for all 4 of those key biotic factors used in the final MCDA model. So more sampling might be needed to complete the picture for every single site.
Okay, data gaps to fill.
Right. And as we touched on with the chytrid fungus, they stressed that future research really needs to zero in on the bead pout pathogen in Sweden, understanding its strain diversity and figuring out exactly which environmental conditions make it flare up and become harmful versus just being present.
Lots more to learn there. So wrapping this up, this deep dive really shows that eDNA, especially with metabarcoding, is far more than just a is it here or not tool.
Oh, much more.
It's a
Remember how the researchers found the chytrid fungus only in the man-made ponds? Their hypothesis was it might have arrived on contaminated digging equipment used during habitat creation or restoration.
right, that possibility.
It raises a really profound and maybe slightly uncomfortable question for the conservation community globally. What might be the unseen role of our own conservation infrastructure? The very tools we use, the boots we wear, the machinery we deploy to help in accidentally spreading diseases like BEAD around the world.
Wow, that's a heavy thought. Are good intentions inadvertently causing harm sometimes?
It's something we need to consider, and it points to another potential future use for eDNA. Not just monitoring the wildlife, but maybe using it to better monitor the contamination risk embedded within our own conservation activities and equipment to make sure we're not part of the problem.
A really vital area for future focus and vigilance. Thank you for joining us on this deep dive into the world of cutting-edge genetic monitoring and its real-world impact.